Hi, everyone. I'm Gina Vallow. I'm a Presidential Innovation Fellow on Dr. Amy Abernethy's team, and I'm excited to share with you a revised version. If you've been with us for a couple weeks, you've seen some ugly slides that we created, and we've really worked hard to put the story together for you all in concert with my colleague, RJ Andrews, who I credit with the amazing artistic work here. And I'm excited to share with you a story. We're going to take an adventure together. This is a story about a single person who's had a lot of COVID tests. This is Heidi. She's had a tough couple of months. We're going to trace her story from left to right around the circle. And we're going to start back here in April when Heidi wanted to get tested before visiting her grandmother. So she went to a nearby superstore drive-through to get tested. Then in May, Heidi was scheduled for an outpatient procedure, so she has a screening at her primary care physician for that procedure. In June, Heidi starts to develop some mild symptoms of COVID-19, so she goes to urgent care where she's tested again. A few weeks later, she develops more severe symptoms. She goes to the emergency room where she's tested and is then admitted to the hospital where she's tested a few more times. And then, thankfully, Heidi recovers and she wants to help others by donating plasma. So she decides to get tested for antibodies. She goes back to her primary care physician to get tested, and she's tested twice after an initial negative result. But this story isn't about Heidi. This is a story about Heidi's data. And at each step along her journey, her interactions generate a mix of structured and unstructured data. And what you'll see throughout this presentation and throughout the story is that we use this filled-in circle to symbolize the completeness and the usability of structured data. So think like a test result of positive or negative that's structured in a field. Versus an open circle to indicate unstructured data, think a clinical note or a message. So we're going to go deeper and take a closer look at the data generated from Heidi's second test. This was when she had an outpatient procedure performed at her primary care physician's office. And here, each circle represents a single data element. And there's only 10 shown, but we know there's a lot more than this in any given interaction. But just for illustration's sake, the green circles here are representing the data that originates in the EHR. The dark blue circles are data that originate in the lab information system, and the light blue circles are data that originate on a testing device or instrument. You'll also notice the red exclamation point, which shows data that's not captured at all at any point along the process. And the important takeaway from this slide is that the data are created at every step in the testing process. And some, but not all data will move between systems. And as it moves, it can shift from being structured to unstructured. Now we're going to take a look at what this interaction looks like on Heidi's bigger map. So if we look to reorient you to this image here, we now have in this green area here, this green shaded area represents the EHR, then the data contained within the EHR. The middle section here, the lab information system, and the outer section or the outer ring of the circle here is what is represented by the instrument or the device. If you notice, if you move outward from the outside of the circle in, you're going to see that data starts off very structured, and we begin to lose some of that fidelity as we move from the machine to the lab system to the EHR. And we also see that by the time we get to the EHR, we have the majority of the patient data, but a lot of it is quite unstructured. Now we look across all 9 of Heidi's tests, and we see differences in patterns and movement of data. And they're all changing based on various providers and labs and devices at play. Nothing looks the same. Heidi has left a messy trail of data behind her, and there's no single system capturing this full story. If we zoom in, we can see that Heidi's primary care physician can see, and in that EHR, we can see three of these tests, and we can connect them together at that level, but that's only a piece of the story. If we look at Heidi's urgent care experience, we get one piece of the puzzle. And if we look at the hospital, we get another subset of results. And even if we had data from all of the EHRs in the green section, we're still missing the part of the story that's happening in the blue section, in the labs and in the instruments. And if you're not overwhelmed yet, Heidi is just one patient. There are millions of Heidis, each with their own messy arcs of data. And this, of course, is the interoperability challenge in a nutshell. It's one many of you on this call are very familiar with and you deal with every day. And some of us might say, this is the argument for the universal patient identifier. This is why we need this to connect the dots. And that's true. It would definitely help if we could connect the data that easily. But it wouldn't change the fact that the data are not structured and that sometimes the data are not complete. And so we have to ask ourselves, what do we do about it? What do we do with this mess that we've created? We think the answer is that we need to start small. We need to start with one data element and figure out how to get that data element to move, and then rinse and repeat. And as part of Project One on the parallel analysis, we've chosen a starting point with the manufacturer data element, which is what we're going to take a look at next. So here, this is an example, a made-up example of what the test manufacturer data points might look like today. And you'll see on the very outside from the instrument, we know this information is fairly structured in the context of a test and of an instrument. And as it moves through, in some cases, it stays, but say in the drive-through, there is no EHR. In some cases, we have it moving to unstructured data, and in some cases, it's missing entirely. And this is really the problem that we're trying to solve for initially. And so we start thinking about, what could this look like? So imagine that we are able to capture this data point from the instrument and move it through the system while maintaining its integrity from the machine to the lab information system to the EHR, and especially doing this using the data standards that are available to us that many in this community have worked really hard to get in place. We'd be able to use data aggregators, you see down here, which we've added into the picture, to start to connect these dots on a patient level. We know that technology exists and we can take advantage of it. If we could do this for one data point, we can probably do it for more. And if you imagine that we could collect data on the instrument, perhaps about the materials used, as we would need to do in the case of Saliva Direct. Or imagine that we could see the cycle threshold that Anne talked about, that CT value for each of these tests, along with Heidi's symptomology at every step along the way. This is what we mean when we talk about connecting the pipes. The ability to connect these pieces and to do this is critical, and to be able to use real world data as a means for understanding the performance and the limitations of the testing available to us. And we've talked in previous meetings about why that is so important relative to the work we're doing on the therapeutic side, on the and on the vaccine side as well. And lastly, I just want to say we're really excited about working with the Reagan-Udall Foundation and Friends of Cancer Research as they kick off the first group of parallel analysis partners. We have a great group of folks that have raised their hands to help solve this problem. They are at all spectrums of this rainbow in terms of stages, and we're all very excited to work through some of these challenges together. And with that, I'm going to turn it back over to Susan to share more about the participants and the parallel analysis.